Measurement or the estimation of disease resistance in the field has both academic as well as practical importance. That is why estimation of disease resistance in the field is the part and parcel of any disease survey program. The information generated from this estimation help us to understand how much disease is present in the field. It also gives us an idea that how much plant or the variety present in the field is susceptible or resistant against a particular pathogen. And finally, on the basis of this data, we can recommend or develop the management strategies. So any plant pathology study actually started from the field where the pathological problem is present, and we can't move forward if we don't know how much our plant present in the field is resistant or the susceptible against the prevailing pathogen. Disease is measured in terms of intensity, and the intensity of the disease is measured by two parameters. One is known as disease incidence and other is known as disease severity. Both are measured in terms of percentage. Disease incidence can be calculated by dividing the number of infected plant with total number of plants assessed, multiplying by 100. For example, if you visited the field and you assess the 100 plants, and out of these 100 plants, you found that there are 70 plants which are showing the disease symptoms. It means that the disease incidence is 70%. Disease incidence is usually measured in the early part of epidemics or those diseases which totally kill the plants, for example, wilt diseases. While when we talk about those diseases which partially kill the plants, like leaf spots, usually we measure them with the help of disease severity. So disease severity can be measured by dividing the portion of the plant tissue damaged by the pathogen with the total area of that plant tissue and then multiplying with the 100. For example, if there is a leaf spot and it is killing 15% of the total leaf area, then the disease severity is 15%. Disease severity or the infection index is equal to the sum of all disease rating divided by the total number of rating multiplied by maximum disease grade and then multiply by 100. This formula is used in order to measure the disease severity or the infection index. There are two methods which are commonly used to achieve or to measure the disease severity index. One method is known as standard area diagram and other is known as descriptive keys. What is the standard area diagram and what is the descriptive keys? How it works in this formula we are going to discuss in the later part of the talk. In case of standard area diagram, you are provided with the picture and that picture shows the different reaction of the plant against the pathogen. For example, here is the standard area diagram of the plant when they are attacked by the rust pathogen. If you look at on your left side, there is a green leaf where there is a no pustule of the rust, and such leaf is known as immune, and they give it thus rating scale 1. And then there is the presence of the pustules which now increasing their size as well as the number, but still the most part of the leaf is green. So if the plant in the field showing this kind of reaction, then it is known as highly resistant. And then the pustule increase in their size and number, and a significant part of the leaf is actually killed by the pathogen, and they give it rating scale from 4, 5 to 6, and such leaf variety is known as moderately resistant. And then finally susceptible to highly susceptible, where the rust almost kill the leaf, and there is complete coverage of the pustules. So in this case, you are provided with the standard area diagram. You can take it in the field and compare the leaf you are observing for the presence of rust disease with them, and wherever you find they are matching, then give it score. It may be 1, 2, 3, 4, and from that data you can calculate whether your variety or the plants present in the field are immune, highly resistant, moderately resistant, susceptible, or highly susceptible. So this way of measuring the disease severity is with the help of standard area diagram. Then comes the descriptive key. Here is the descriptive key of the white mold of beans. From the descriptive key, you can get an idea where the description of the events is given. For example, if we look at this key, there is a rating scale 1. It means that there is a no disease. For example, if you went into the field and on the bean plant you can't find any kind of disease, you score your plant in the 1 category. But if you find 1%, around 1% of the area of the plant is damaged by the pathogen, then you score your uh, plant in the 2 category, and it means that the trace of disease is present. And then if the damage is from 1 to 10%, it means light disease is present. And if the damage is between 11 to 15%, then moderate disease is present. And if it is more than 50%, it means that severe disease is present. And you can just in your lab book or in your field book score the plants according to this rating scale, and then finally calculate it. So in the next part, through example, we are going to understand how disease severity index or infection index is measured. Suppose you want to calculate the disease severity index or the infection index of the bean plants which are infected by the white mold, and the total number of plants you assessed are the 100. So these plants vary when it comes to the disease severity. For example, out of these 100 plants you assessed, 10 are the plants which are not showing any kind of disease symptoms. So you give them disease grade 1, and the number of rating is actually the multiple of the total rating and the disease grade. If 10 multiplied by 1, it is 10. Then there are the 20 plants which showing the disease, but just around 1%. So you give them disease grade 2. And then there are the 50 plants where the damage is between 1 to 10%. And then there are the 10 plants where the damage is between 11 to 25%, and according to that you give them disease grade 4. And then there are the 10 plants where the damage is more than 50%, and then you give them disease grade 5. So the total rating is 100, and the number of rating is the multiple of that, it is 330. So sum of all rating is 330, the total rating is 100, and maximum disease grade is 5. If you look at the disease grade, you can find the maximum disease grade is 5. When you put it into the formula, you get that the disease severity index is a 66%. It means a severe disease is present in the field and which can lead to the crop failure. Or in other words, our bean plants present in the field are largely susceptible against the white mold of the bean caused by Sclerotina sclerotium. So on this basis, you can recommend that the measures must be taken to control this disease, and because the disease is present in the field, the new research must be initiated so that the future crop remains safe of the pathogen. This is how by measuring the disease in the field, we can assess whether the variety or the plants present in the field are resistant or the susceptible, and that data is published and accepted by the scientific community all over the world. Thank you.